So today my lecture will be close to the topic of this conference. So hopefully it will be helpful for all of you, great mathematicians. So I will talk about neuronal dynamics in neurophysiology. So dynamics is, the word dynamics is used in various ways in neurophysiology. So the most general meaning is that this is something changing in time. But more recently, neuronal dynamics started to be used in some specific way, and I will talk about this. So if you look at the history of neurophysiology, which probably starts from the work of Evers, and you just look at the pictures that they had in the papers, then you see that over the years, over almost 50 years of this research, the pictures have been changing. So like in the early work of Evers, you see these post-stimulus histograms, which represent activity of single neurons. Then with the introduction of population approach by Georgopoulos, these figures started to change. Then Nicolelis and his colleagues introduced multi-channel recordings, and then the pictures became more colorful. And now we have this neuronal dynamics approach where instead of neurons, peristimulus time histograms or waveforms, we see states and some state transitions, rotations, which look beautiful, but sometimes this stuff is hard to interpret. So let me just overview the history of this a little bit. So traditional neurophysiology started with Evers, who started single unit recordings in monkeys. So the monkey was awake and it was performing some motor task like pull or push a lever. And Evers would draw pictures like this. So this is the central line is movement onset. And then this is a peristimulus time histogram, which represents the change in the neuronal rate as a function of time. So if the monkey pulls the lever, then the activity of this neuron increases. If the monkey pushes the lever, the activity of this neuron decreases. This is a raster display, and here each line represents a behavioral trial, and dots represent single unit discharges. Notice also that these discharges are very variable. So then came Georgopoulos, who enriched the motor repertoire of the monkey, and the monkey would be moving the hand in different directions. And Georgopoulos described this directional tuning curve, where on the x-axis you have movement angle, and on the y-axis you have neuronal firing rate. Directional tuning is an important concept in neurophysiology, and typically a neurophysiologist would say that a neuron represents movement direction. There is a preferred direction. For example, for this neuron, the preferred direction is 180 degrees because it fires the most when the monkey moves the lever in this direction. Also, there is a variation of this task. It is called the instructed delay task. Here, a monkey or even a human is given an instruction of movement, like move in this direction, but then he is not allowed to move until there is a go cue. So when the subject knows movement direction, but is not allowed to move, there are still changes of activity in cortical cells. For example, this one responds with a brief response to the instruction where to move, and then the activity slowly grows till the time the trigger command to movement is given. Also notice a high variability of this neuronal discharge in single trials. And finally, I will show a similar example from my own work. Here, a monkey was pressing a button and then mentally counting 3 seconds. So it was measuring time. So the monkey was overtly not doing anything, but it was, you can see that this neuronal activity increased as the count of time proceeded. Again, notice that there is a huge variability in these neuronal discharges, which may carry actually a lot of information, but unfortunately, we do not know what kind of information could be contained in these neuronal discharges. And most people look at these peristimulus time histograms, at the changes of average firing rate, and this is our current situation, also with the more recent neuronal dynamics approach. A few more words about directional tuning and preferred direction. It appears that it is not fixed, like one cell prefers movement to the left. What do I mean by this? Here, this is a little bit complicated slide. It describes activity of a single cell when the monkey performs something with the hand and then with the brain-machine interface. But let's just look at the case when the monkey performs movements with the hand. So if you look at time 0, there is some directional tuning, in this case 45 degrees. But if you move the analysis window back and forth, then this directional tuning preferred direction rotates. And this has been demonstrated by many authors. So basically, preferred direction is not very fixed, and it could rotate depending on where you look in this neuron's activity, like before the movement, after the movement, when the monkey prepares the movement. So it could be that preferred direction is not the best description of neuronal activity, but it is still useful. The next complexity came with the neuronal population recordings, and they were probably first advertised by Georgopoulos, who proposed a population vector approach. So in this population vector approach, each neuron's preferred direction is detected, and then this vector pointing in the preferred direction is multiplied by the firing rate. Then a sum is taken, weighted sum is taken of these individual neurons' preferred directions. And then with this approach, if you have many neurons recorded simultaneously, then you can predict movement with good precision. Even though in the early work, Georgopoulos proposed mostly intuitive approach of weighting these vectors, so it was not optimized, like multiple linear regression was not conducted. But still, when he combined many neurons, then the precision of this population vector prediction improved. So this was a population. But there was a problem with this. The problem was that Georgopoulos did not record all neurons at the same time. Instead, he recorded one cell at a time, and only later, maybe 6 months later, he could construct this curve. So this is not ideal. One would like to record many neurons at the same time. And this has been done. So early work dates back to something like 1970s. Humphrey et al. Recorded maybe 5 to 10 cells and simultaneously, and then they used a linear regression algorithm to predict wrist movements from neuronal activity. So this was probably one of the first use of linear regression and simultaneous recordings of many neurons. Then in the 90s, Miguel Nicolelis and John Chapin increased the number of simultaneously recorded neurons because they implanted many electrodes in the red brain, and they recorded several tens of neurons. So the number of the neurons increased, and also the predictions of movements improved with this approach. This is from my own work. Here I recorded 394 neurons simultaneously, and I'm particularly proud because these are good recordings. So on most channels you see clear single units. Of course there are multi-units as well, but many of them are single units. Nowadays people resort to multi-unit recordings. So if you see a paper with multi-channel recordings, like recently Elon Musk demonstrated a pig with many electrodes implanted. So I'm pretty sure that he in this pig he had multi-units. But in any way, one can record a lot of neurons at the same time. So, and with all these recent developments like Musk's Neuralink, this will be developing more and more. And with more neurons, then the diagrams that the diagrams in the paper improve, they become colorful. So also in my paper, this represents recordings from around 100 neurons in the motor cortex, and I sorted them by the time activity peaks, and you can see that there is like a wave of activity running across the motor cortical neurons. So remember this wave because I will show it once again later. Also, since we are recording nowadays many neurons at the same time, then we can think about the principles of neuronal ensemble physiology. This has been published, and I recommend looking through these principles. The most important one is distributed coding, meaning that single neurons are interesting, but the most interesting stuff comes from the population. And the advantage of the population is typically described as a neuron drop-in curve. So the more neurons you have, then the better the quality of precision. And actually, until recently, it was around maybe 50 or 100 cells. And then some people got a false impression that neuronal drop-in curve saturates when the population reaches the size of 50 neurons or so. But that was a false impression because these people did not understand statistics very well. The problem here is that if you have a limited sample, then when you play with this sample at the number close to the sample size, then you cannot really say anything because whatever you do, you get pretty much the same sample of neurons. If you increase the sample of neurons, let's say to 5,000 neurons, then there is no saturation and the curve continues to grow. Of course, still there is a problem because to get some reasonable improvement in decoding, you need to go to the next order of magnitude. So you have 10 neurons, this is good. then you need 100 neurons, then you need 1,000, 10,000, and so on. You need to grow the number of channels exponentially to get a good prediction. And finally, with the population, you can look at directional tuning sensitivity in many neurons simultaneously. So this is a little bit of complicated curve, but the take-home message here is that when a monkey performs something by moving the hand, you have a diversity of preferred directions. When you switch to brain control without movement of the hand, everything changes very dramatically. So, and then it takes a monkey several days or weeks to get back to normal when using a brain-machine interface. So this was the situation until recently, and then neuronal dynamics came. And the first paper was on rotational dynamics. This paper by Churchland and his colleague, the last author is Krishna Shenoy. And this paper was published in 2012. And it became very, very popular. It gets probably 100 citations per year or maybe more. And basically, these authors proposed, as they advertised, a revolutionary approach to looking at these things. So they basically said that all previous people that talked about neuronal representations, like a neuron represents direction or the neuron represents force, etc., etc., this is all incorrect. And instead, you have to look at the neuronal population as a dynamical system. So, which sounds as a good idea, and let's look more closely at the paper. And they conducted some manipulations and reported that if after this, if you do these manipulations, you show that neuronal population response rotates. So in their case, the monkey was doing some kind of reach in different directions task. Sometimes the monkey avoided obstacles. So the movements were similar, but also different in many aspects. Then they did PCA analysis and then manipulated matrices and forced the activity to a plane where rotation was visible. And very impressively, these rotations were pretty much in the same directions for all trials. So what is rotation in neuronal space? This is a circular movement of some kind of vector pointing from the center of coordinates. And intuitively, it is clear that if one neuron passes activity to the other, so one neuron, then the other, and so on, then you would likely to see some kind of wave traveling across the population. Unfortunately, although this was intuitively clear, I didn't find this wave description in the original paper. Instead, they talked about oscillations and some very, very surprising results. So, which made me take their data, it was available online, and try to reanalyze it. So here, this is their data. Y-axis represents neurons. This is time. By condition, they mean different movements. So like movement to the right, movement to the left, or movement to up, but through some convoluted trajectory. So different movements. So first of all, and also here notice that I normalized activity from -1 to 1. simply not to look at the differences in the firing rate, but to pay attention just to when peak activity occurs. And so when I averaged all trials and sorted them by peak activity, there was a nice wave of activity running across this population. This is actually not very new. I just showed it from my own paper, and other people observed these waves. But what is interesting here is then when I ordered these neurons the same way as here for different conditions, then you can see the same wave. So basically, whatever happened in their experiment, their conditions were similar enough, and there was an orderly fashion in which these neurons were activated. This one was the first, and this one, and so on, with some variations, of course. So if you look closely, you can find a neuron that exhibits very different activity in different trials. But still, this activity wave is pretty obvious. Also, you can just calculate activity peak in different trials, and then plot them, and then once again you see this wave. So since this wave was present, I played with the data a little bit. So I split the population into early activated cells and intermediate activated cells and late activated cells. And then I just plotted average activity of these three populations. So early, intermediate, late, for different conditions, and very clearly for all conditions, early cells were early, intermediate cells were intermediate, and late were late. You could even shuffle conditions, and then still this was true. I could plot this in three dimensions, and then this rotations was clear and it did not disappear when I shuffled conditions. So here, without doing any advanced mathematics, I could easily obtain these so-called rotations, and they clearly came from the sequence of activation in this population. Then I actually did PCA, and then if you do a PCA, then you plot the first principal component, second, third, then you observe rotations. And if you shuffle conditions, then peculiar features of different conditions disappear, but still there is a sort of rotation. Now let's compare this with the analysis of Churchland and his colleagues. And here it is called JPCA, and I will not go into all details, but it is a sequence where they first pull out the 6 principal components, then they rotate matrices, so just to pull out just the rotations. So if you do this analysis, then you get these rotations. Then what I did, I shuffled conditions like here, but then all rotations disappeared in JPCA. This was a little bit surprising for me, so I didn't know what to think, but a little bit later I will tell you why this happened. But in the meanwhile, I did a very simple simulation where I assigned to each neuron a response that looked like this, and I randomly assigned response amplitude to different conditions. The only requirement I had was that these neurons, simulated neurons, were activated in a temporal sequence. Now, when I did this and applied JPCA, then I didn't get any rotations. So I wondered why I'm not getting these rotations here. So this is strange. Maybe this maybe some matrices get badly conditioned or something like this. So what I did was splitting the population in half and making this wave in half of the neurons start early, and then in the second half the wave activity started with a shift. So this didn't change the main idea of a wave running across the simulated neurons, but it improved the matrices, what I thought. So, and after I did this, then these rotations immediately appeared in the analysis, so I was kind of happy. Then I simulated activity where there was no temporal sequence in neuronal responses, and then the analysis didn't return any rotations at all. And what I did next, and actually I asked Alexei Sachi to help me with this, just to look at their code, and he looked and said, wait a second, I will tell you what is happening here. Like here, when this rotation disappears for shuffling cells, this could be related by one trick that they do. So they take an average peristimulus time histogram for all cells, and then they subtract it from individual cells. So this is like a questionable manipulation, in my opinion, because why subtract some kind of average activity for different conditions from a particular condition? But in any way, this manipulation really can kill rotations for some cases. So if you do not do this subtraction and just apply the method to the activity, then you get clearly rotations, and also you get rotations when you apply it to the shuffle data. So this is there, shuffle data, all conditions are shuffled, apply the JPCA without this questionable subtraction, and then there is a rotation. So now it became very clear to me that if there is a temporal sequence in activity, then you apply this method, you get such rotations. Then I asked, so like if a goal is to obtain rotations, but let's just obtain these rotations directly. So let's fit the data to a circle or to a Lissajous curve, which is an 8 or infinity sign. And then it was very easy to fit their data to pretty much anything. So you can get a circle or an 8, or you could even get this prediction. So train on one part of the data and then apply to the other, and it would predict circles or 8s. So which means that probably just when you have a goal of getting something like rotations or any shape, then you should have a better idea why you are doing this. And okay, so finally, where to read about this? You read about this in our paper published in Scientific Reports. And there was also a recent review by the original authors, and they actually responded to our paper. But I was actually really surprised how they responded. So first of all, they claimed that we demonstrated rotations only for simulated data, but empirically you observe something else. This is strange because I mostly use their own data to discover the sequence of responses. And next, they said that rotations are something else compared to the presence of sequential activation, because the neurons are typically multiphasic and have peaks at different times for single neuron responses, etc. So, and referred to the figure number 2, and indeed they picked out like 1, 2, 3, 4, 5, 6, 7, 8, 9 neurons out of 200, and they focused on some changes across conditions. But I don't know what they mean by that, because I just showed you the whole population of 200 cells, and this wave of activity is clearly present there. So probably I will have to write some kind of response to them. And lastly, I will mention the neuronal subspaces idea, which also comes from the same group. And what they did, they looked at a representation of movement by neurons during the movement and during the delay preceding movements. And they described the transition of neuronal activity N into movement X as a matrix multiplication. And then they looked at the elements of this matrix and introduced a term of neuronal subspace. So basically, neuronal responses for movement are confined to some subspace and neuronal space. But if you look at neuronal responses during the delay, then this activity is rotated 90 degrees from the movement-related responses. So, and they came up with this nice interpretation that the reason why there is no movement when you prepare it, but there is activity in the cortex, is because all neurons are active in a different subspace. And then to produce a movement, you just shift to a new movement-related subspace. So this is a very nice and sounding reasonable idea. The only problem is that before them, Georgopoulos looked at the same problem, although he described this slightly differently using a population vector approach, but still he used pretty much the same metrics. And but in his case, there was no 90-degree rotation between the delay activity and movement related activity. So which made me look more closely at the experiment, and it appeared that in the Georgopoulos experiment, the monkey was free to look anywhere. So the monkey received an instruction and it can start to look at the target with its eyes immediately. But in the Kaufman experiments, the monkey was forced to look at the sensor all the time. So they had this condition where the monkey was looking at the sensor, but preparing a movement with the hand to a different position. So which may have actually contributed to this 90-degree difference between the delay activity and movement activity. So, but still, this subspace idea is interesting, and this is something to look at in the future. So I'm done. Thank you. So if you have any question, I will be glad.
Thank you very much, Mikhail. Any questions? Well, I have a question.
Okay.
Actually, I have two questions. One is
Yes.
technical. When you are talking about shuffled data, how do you make shuffling? What statistics do you preserve and what is changing? It was a shuffling between channels or shuffle?
Yeah. So in this case, it was a shuffle across just conditions. So just movement. So let's say for a given movement, there is some responses of different cells. And you keep this cell, but take a response from a different condition. So say monkey moves to the left, I take some other trial and then insert the data when the monkey moved to the right.
Okay.
So this basically removes the contribution of what the monkey is doing and leaves just a general pattern of activity in the population. But there is no shuffle across cells, so just conditions.
And the second question is, say, more philosophical. When you set up this recording matrix into the brain, can you stimulate using the same electrodes, recording?
Yes, absolutely.
And can you invent a stimulation pattern to set up the hand in particular position?
Yes, absolutely. This can be done. So and this hasn't been done for motor cortex. So this is something, some interesting experiment to do in the future.
Okay, thank you. Really intriguing. Okay, anybody, any questions, any more questions?